Alrighty, good evening, ladies and gentlemen. Thank you so much for coming. Hi. Hope everyone has found plenty of pizza and food and snacks and all these healthy foods. If you haven't been to a Young Adult Science Cafe before, welcome. This is a monthly event that we hold. And the idea is to not only inspire the younger generation of soon-to-be scientists and healthcare professionals, but we also want to reveal the realities and the limitations of a career in the sciences. When we're younger, we have these ideas of what it means to be in science, or what it means to be a doctor, what it means to be a research professional, what it means to get a PhD. But at that age, we very rarely consider the true trial and error, the true cost, the true investment. So the main goal here, the main target audience for the Young Adult Science Cafe are the younger students in the audience. So please feel free to ask questions throughout the event if you have any questions whatsoever. So today we have an excellent gentleman, Dr. Peter Billingsley, who works for Snanaria Inc, if I'm pronouncing that correctly. Almost. Excuse me. Yes. They primarily work on vaccinations for malaria. Most of you are quite familiar with malaria. I'm sure you've heard of it. This gentleman has quite a bit of experience. He holds his bachelor's in sciences, PhD in biology, and has a very impressive resume. So I'm quite confident that he has a lot to say. He will be discussing primarily, as our science topic for today, as you can see, the developing and testing of whole organism vaccines against malaria, and then of course we'll wrap it up with a brief overview of graduate education, perhaps advice, the realities, the limitations of pursuing such a career. So enough of me talking. Thank you all for coming. If you could please join me in welcoming Dr. Peter Billingsley.
Good. Okay, can everybody hear me all right? Thank you, Caleb. I'm going to actually, can we get the music off? I can still hear it. Oh, it's from outside. Oh, what's the party outside? Is it Freshers Week or something? All right, okay. No worries.
There's no frequency.
There's no frequency? Can you hear me without this? People at the back, without your mouth full of pizza, can you hear me with this? Without this? Or do you need it for recording?
Oh, no.
All right. Okay, good. So I'm going to do things in the reverse order from what Caleb just introduced, partly because I didn't know he was going to introduce it that way, and partly because I already had the slides prepared from a couple of years ago and I've modified them. So what I'm going to do is, first of all, tell you how on earth I got to where I am. And you can ask questions along the way and so on. But how did I get from this? And I tell you, I'm the handsome one on the left here, not the ugly one in the middle or the smiley one on the right, to this. And again, this is me at Snanaria, and again, I'm the handsome one on the left. Steve won't be very impressed if I tell him that, but there we go. So what I want to do is just try and give you an idea of how things worked for me, and you can ask questions and how these things happened, but let's just go through a little story. And some of it's about unexpected events, some of it is about people, a lot of it about people, some of it is about opportunities just to rise at a certain time, and a lot of it is about just go for it. Don't worry about things, they'll nearly always work out, and if they don't, you'll get another chance. So people, first of all, way back when I was your sort of age, this man here was my high school teacher in biology. His name is Spike Walker. He's probably about 85, 90 years old at the moment, and he was a brilliant microscopist. It was his hobby, it was his passion, and he had microscopes and actually enjoying it when I was 16 to 18 years old. He's very active, and he 2 years ago won a prize for one of his microscopy pictures from an institution called the Wellcome Trust, which is the biggest charity funding medical research after the Gates Foundation. So Spike was very inspirational. He was also very funny. And all the time that I was doing this microscopy and enjoying things, I was thinking, you know what, I really want to be a vet. I want to go and work with animals and do all this stuff and push my hands up a cow's butt and all that sort of stuff. It really appealed to me. But while I was 16, 17, 18 years old, these books came out. These are a guy called James Herriot, who was a famous vet, made movies about his books, had series on the television every week that everybody was watching and talking about. And guess what? The application rates to vet school went from here to here. And it became uber competitive. And I didn't get into vet school. And so I ended up sticking with the animal theme and with sciences, and I went to Leeds University, which is in the north of England. It doesn't look like it's very much in the north, but remember Scotland starts somewhere around here. And I went to Leeds University, and it started a bit of a theme for me, which is that I ended up in places with very ugly buildings. This was our building. I did a degree in agricultural zoology, and this was called the Ag Zoo building. And we had our final year research projects in this end classroom here with windows way high, glass about wafer thin, and in the middle of the winter we were wearing our coats in the lab and putting gloves on with our lab coats on top and so on. It was horrendous. But the rest of Leeds University looks something like this series of concrete monstrosities that afflict many of the universities all over the world, I'm afraid. But the man, again, I'm talking about people here, the man who really mentored me very well and gave me great sage advice when I was at Leeds was this man, Donald Lee, who was studying worms. He did a lot of work on worm populations and worm biology, particularly in Brazil. He spent a lot of time in South America. He told us gruesome stories about deworming whole villages and seeing the effects in the open sewers a day later and so on. Sorry, you're eating. But Donald was a magnificent professor, really, really solid man, very quiet and very, very English and British, and offered me a PhD, and I turned it down because I didn't want to stay in the same place. And when I turned it down, he said, look, that's all fine. He said, one word of advice is don't look back, don't go and have regrets. Now this is something that could really put you off your food. I'm sorry. This is Margaret Thatcher. And again, at the end of my first degree, things went a bit awry. Margaret Thatcher was in power, and she completely slashed the education budget for postgraduate education. My dream PhD at the London School of Hygiene and Tropical Medicine was slashed in that same time. So I didn't get to go and work on sleeping sickness, and I was in a bit of a bind. And in August of whatever year it was, 81 I think, 82, I got this phone call from Canada to where I'd applied, and my future supervisor said, why haven't you responded to my letters? Where are you? Are you coming or not? What the hell are you talking about? I don't know this man. And it turns out there'd been a postal strike in Canada, and my application letters had been thrown in a river in Ontario somewhere. But a few months later, here I was in Canada in another god-ugly building, but enjoying a PhD. And so what I'm trying to convey to you here is if you get so uptight about how things are going to work out, am I going to get into the place of my dreams? Am I going to do this? Am I going to do that? It doesn't always work out, but it's always... Yeah. Okay. Is that better? Okay, good. Yes, things will work out. And I ended up doing a PhD, spending summers sometimes at this little field station on the Rideau Canal, north of Kingston, Ontario, which is right here at the end of Lake Ontario. And since we moved here, I've had a fun time going and seeing old friends there after many, many years. Mentored by this man, who is possibly the funniest academic I've ever come across. But he was the man who, first of all, got me really, truly interested in entomology. and in mosquitoes and so on. But he also had a magnificent ability to convey complicated science to people who wouldn't otherwise get it, particularly in the area of biochemistry. He would get extremely excited about a hydroxyl group being stuck on the side of a molecule, which bored the pants off me, but suddenly he could make it exciting and say, oh, you've got to just change this and such happens. You end up with a completely different active molecule, and it sits in the receptor there, and he gets all excited about it. And so he was a tremendous mentor for me, and he probably gave me a couple of very important things. One is to be enjoying the science that I do, allowed me to consider that having a sense of humor in a lab and in front of a classroom, in front of situations like this, is perfectly acceptable. Thank you very much. You can have a lot of fun while you're doing what you're doing. And he made it okay to have other things going on in your life. He was a wonderful pianist. His one failure with me was that he tried to teach me to play piano as well, and that's like watching a guy with two left hands. It was rubbish. But he was an excellent clarinet player, and before he got multiple sclerosis, he was a semi-professional basketball player and had played against the Harlem Globetrotters at one point. So please come in. So again, he really showed me that there were a lot of things to do in life other than science. And during that time, myself and my wife, who's sitting at the back there, spent a lot of time playing music in public. It helped to pay for our bread and butter a lot of the time. And I played a lot of soccer at that time. But this was the beast that I was working on. This is one of the beasts. This is a bug called Dipetalogaster maximus. And as you can guess from the Maximus title, it is the biggest blood-feeding insect. It's not as big as a tick, but technically ticks are not insects anyway. But this thing, when it feeds, it takes about 2.5 mL of blood in one go. And then it sits in a little crevice somewhere and digests it. These things normally feed on lizards in desert environments, so they're very good at retaining water and so on. And that really fired me up. I wanted to go down to Latin America and work on Chagas disease and so on, but Al Down, my supervisor, said, no, this is a lab project, hard luck. And then during that time, I was lucky enough to be offered a postdoc to go to the Swiss Tropical Institute in Basel, this wonderful little building, which will soon be abandoned, and they're building a big new one somewhere else. And this gives me a nice segue, actually, because sitting at the front here is Jongo, a very dear friend of mine from Tanzania, and he's just started. So Jongo is one of our lead clinicians in our work in Tanzania, which you'll hear about a bit later, but he's just started a PhD in Switzerland at this institute. So we went all the way across the world, from Canada back to Europe to tiny little Switzerland. And one of the provisos for that move was that the Swiss Tropical Institute were going through a refocus. They said, we really want new people coming in here to work on malaria or something related to it. And hence began my love affair with mosquitoes, which still goes on. My love affair is a bit much. But there was another influential person in life here who is actually going to be, is he your supervisor? Yeah, he's going to be Jongo's supervisor. And this chap, Marcel Tanner, became the head of the institute for something like 25, 26 years. Took it from a tiny institute to a major international research organization. But a lot of it was founded upon the institution that Jongo is from in Tanzania. And again, my work was lab-based, but meeting Marcel, he said in his very Swiss voice, Pete, you have to go to Africa, you have to go. If you really consider yourself to be someone who wants to work on malaria, you've got to go there. And I had no quibble with this at all. It was just what I wanted to do. And it was Marcel made that happen. But before I went to Africa, I went to Imperial College in London. This was a big fellowship that I got from the Royal Society, and my office was down here somewhere. Very nice, because the pub is right here. And interestingly enough, my youngest son hangs out there more than here. So Imperial College was a great, it's ranked always in the top 10 universities in the world. And it's a tremendous place to spend some time. It served me very well. But because I had this fellowship money, I could go from London and I could set up my own research program. And I decided to continue with what I'd been doing in Switzerland and start to look at malaria transmission through the mosquito. And I got involved in many different projects at the molecular biology level, at the ecology level. At mosquito physiology was where I gained my training from Canada and then through Switzerland. And it all came together very nicely for a few years in Imperial College, and that's when I started going to Tanzania. And this is a picture of a little boy with his mum looking out over the rice fields, just about a mile away from where this picture is taken. There was a house where my friend and I used to go and sample mosquitoes every night in what's called a light trap, and we could collect up to 10,000 mosquitoes from one house in one night. 10,000, right? I mean, it doesn't bear thinking about now. I mean, that's not like that anymore, and nobody's quite sure why, but we would go back 5 or 6 nights a week and get 10,000 mosquitoes. I can see people starting to scratch at the thought of this. It was, and you can imagine just how intense the malaria transmission was in a place like this. When we started, we could go at any time, and 85% of the people would have malaria in their blood. So it was like the biggest problem, and working on the biggest problem of all from the mosquito front. It was a real joy. A lot of good work was done at that time. I'm only putting this up because I like the photographs that I took, really. But you can see the conditions people lived in. John and I were talking about this earlier, that a lot of the time people don't get to enjoy their lives in places like this, right, John Go? Because they have to work so hard. They have almost nothing. And this was somebody's kitchen. You can see these kids out here. There's rice fields around them, and they'll get bitten and bitten and bitten by mosquitoes until they go to bed. And after they've gone to bed, those mosquitoes just keep going. So it's a huge problem. We did a lot of work doing things like giving out bed nets, as you can see on the left there, developing new trapping devices that we could take anywhere that we wanted to in the area. And so that became a really important part of what I was doing, and enjoying the field work and relating it to the lab. And part of that relationship was I was trying at the time to develop a vaccine against a mosquito. I wanted to make a mosquito, if it fed on a vaccinated person, it would keel over and die. I still think it can be done. I just don't have the bandwidth to do it at the moment. But that interest attracted people from the Government Veterinary Institute, and they said, how do you fancy trying this with sheep scab? So you've probably heard of a thing called scabies. This is the sheep equivalent. And this is what happens to sheep when they have sheep scab. They get an allergic reaction all the way through the skin. These little scabies mites burrow into the surface of the skin. They leave bacteria behind. You get these secondary infections. The sheep lose appetite. They lose weight. Their skin falls off. The wool falls off. And they die a very slow, painful death. And so we started working with the Veterinary Institute of the government to develop vaccines for this. And that was part of the whole portfolio of things that I took with me when I went to Scotland. That was my next move. I actually took this work to Scotland, and curiously enough, that sheep scab is more important in Scotland than anywhere else in the UK. And again, you know, the University of Aberdeen is actually one of the oldest universities in the world. It's about 550 years old. It's got these lovely buildings. But zoology, the ugliest building on campus. It was horrible. It was really a truly horrible building, but great people to work with, and a real testament to the place that people hung in there. So I started heading further north, and because of that interest in the sheep's gap, it allowed me to go away from just mosquitoes and to go into other areas of work. This idea of having a vaccine against something that feeds on the outside of you rather than lives inside you was something that was gathering interest. And on here you can see these sort of mini alien-like creatures. These are copepods, more closely related to crabs than anything else. And it's called the salmon louse, and it feeds on the skin of the salmon. And eventually it'll graze away the surface of the skin, and those salmon will die of osmotic shock because they've lost the ability to osmoregulate across the skin. And this is a huge issue in salmon farming, and salmon farming is the third most important economical income for the Scotland. The other two being oil and, anybody want to hazard a guess? No. No. Comes in a bottle. Scotch whiskey, malt whiskey. So this is the third most important. And then tourism is the next after that. And with tourism, you have a lot of red deer and some beautiful, beautiful places to work in Scotland, to walk, sorry, not work. And these deer carried a lot of ticks. We could find deer that, we actually worked with people called gillies. These are people who run the, what do you call it? The estates, the big estates in the Scottish Highlands, and they'll cull the deer periodically. And we would ask the gillies to send us a 10 centimetre square patch of skin from just next to the butt of the deer. And in some of those we'd get 200 or more ticks from one deer just sitting there. All very well, but these ticks are transmitting Lyme disease all over Scotland now, and that Lyme disease was spreading as the 10 years that we lived in Scotland. That Lyme disease spread right the way through the country, and part of our work was to understand the dynamics of the ticks in the Highlands. While I was working on all those different things in Scotland, I started interacting with this gentleman, Steve Hoffman, who's the CEO of Snanaria, and he, even before I came here, I'd met him a few times, but he was developing Snanaria, and it was this tiny little company at the time of 7 people. And he had a concept, and he used me as a consultant to come over from Scotland to help grow the mosquitoes in a certain way, which I'll talk about in a moment or two. And so it was all about, again, it's all about people and sharing visions and so on. And with Steve, this whole thing was, this is where we want to be. We wanted to be vaccinating people, we wanted to be vaccinating children in Africa, and we wanted to have a malaria vaccine. So over the couple of years that I was consulting for them, and before I came to work here, it was clear to me that the vaccine that I'm going to tell you about in a minute had a much more chance of making a difference than anything else I was doing. And it felt like a natural move to go to Snanaria and push my efforts there. So that is a potted peat history, if you like. And I think the important things there is what I said right at the start. Think about people. There are going to be people that give you good advice and bad advice, but use it always. Enjoy the people that you're with. I've been in charge of many PhD students in Scotland, and if you've got questions at the end, I can tell you about some of those. But it's very hard to get past being miserable when you're a PhD student. You have to enjoy it. You have to enjoy the people you're with, and if you you're not enjoying it, fix it very quickly, because otherwise it'll be a horrible 3 or 4 years. The same for any degree. I mean, our son has just, last year, just changed degrees completely, and he's gone from being very unhappy at Imperial College to being very happy there. And that just makes a big difference. So you either change it or enjoy it. And so people grabbing opportunities and not worrying about when things go wrong, because they will go wrong. I promise you, they will go wrong. Okay, you'll get disappointments, but it always works out. So that's the first part talking about, and now we'll slip into the science. Before we do that, I need a drink of water. This is what's happening this week. It's the World Health Organization. This week is World Immunization Week. Which I didn't know about until earlier today. But it's interesting, I don't know just how many people here understand about vaccinations in this country, but it still amazes me that there are people who do not want to vaccinate their children against measles, rubella, and so on. It's an interesting set of discussions goes on year after year about this. So let's switch our attention now to the science and to malaria. The main cause of deaths in the world from malaria is from this organism called Plasmodium falciparum. And depending on who does the analysis and how the data are gathered, the number of deaths are estimated to be anywhere between 440 and 730,000 per year. Staggering. It's truly staggering that many people can die in a year from one disease. And about 214 cases of malaria a year. And this is just horrendous. To put it into context, when the polio vaccine was released in the US in the 1950s, I believe, they had about 100,000 cases. And about 20% of those would die in those days. So you're looking at, taking 4 digits off there to put us on a par with polio. Very, very different scale of the problem. Over the years, since about 2000, there's been a big push to improve malaria control. And a lot of that is on bed nets, and that was one of the things I was doing a lot of in Tanzania, was research into bed net distribution and use and so on. And things like bed nets and malaria drugs and spraying for mosquitoes inside houses have brought the number of cases of malaria down by about 50%. Oh, sorry, the deaths, not the cases, by about 50%. Did you have a question? Oh, sorry. But as you can see from this curve, despite all that control, this is scarily starting to flatten out. And there are places that we know of in Africa that people are saying, actually we're seeing more malaria than we did before. In Jongo, I should have got your slide for that really. Everywhere in Tanzania except for 2 districts, I think, malaria has increased in the last, how many years? 4? 4 years. Despite all of the types of efforts that are going on here. And millions of dollars being spent. Now, how has Snanaria got to where it is? It's all about people again. Of course it's about money. We have managed to gather funds from all sorts of different areas. And we are unique, I think, in having African governments spending money on malaria research and vaccine development. But again, it's all about people. Here is a list of institutes with which we collaborate, but in those institutes are lots of people. And every year we have a meeting like this, and there's always at least 100 people. Sometimes it's been up to 130 people. If you look carefully, you'll see John going, oh, there he is. Sitting at the front, one of our prime guys at the front here. And these people come pretty much for free. They come from all over the world. Every one of those people is now working in some capacity on our vaccine, which is pretty, it humbles me a lot. I'm really happy to be involved with this. If we didn't have this sort of participation, these big open discussions, this is another meeting that we had in a library in Germany, where everybody got together there. If we didn't have this interaction, this participation, this group dynamic that is so important, we would be a long way back in our research and development program. We rely incredibly heavily on our friends and colleagues. So what are we talking about? We're talking about a malaria vaccine that is grown in mosquitoes, and we want to get that vaccine out into the market. Because we're a company, we're not like an academic institution, we think about the markets all the time and where are we going to take it, because that's how we gather our money to do our research. So we need a vaccine that we can use in places where there is malaria already, either for mass vaccination campaigns where we've tried and go out and vaccinate as many people as possible, or targeting it for various individuals. So for example, pregnant women are very, very susceptible to malaria. They lose the baby, they get spontaneous abortions, the children are born underweight and so on. It's very, very difficult time for them. And at the same time, we would also like to have a vaccine that will protect people when they travel to malaria endemic areas. And that means people like you and me, it means people like military, people who work in big oil companies and so on. Fortunately, regardless of how you plan to use that vaccine, the characteristics need to be the same. What we need to do is to stop people being infected with the malaria parasite. Not just being better, not just feeling better, but you have to stop it infecting people. And if you do that, you can prevent malaria being transmitted, and you stop people getting sick. So this is how it works. This is pretty much the most technical slide you're going to see today. This is the life cycle of malaria, and if you don't remember any other word, just remember this one: sporozoite. The sporozoite is the stage of the malaria parasite that gets spat out by the mosquito into the bloodstream. Okay? And those parasites go to the liver. They grow from every one sporozoite. You get about 40,000 parasites. Each one of those goes into the red blood cell, and each time it goes through a cycle, it grows another 10-fold. And when they hit the red blood cells, that's when people start to get sick. And this cycle here lasts approximately 2 days. So a classic set of symptoms for someone with malaria is, after this time point here, which is about 5 days, they feel a bit lousy. Then they feel better. The next day they feel even more lousy. They might have a bit of a fever, a bit of a headache. Then they'll feel a bit better the next day, and then they'll get worse and worse, and it's like an upward spiral. And eventually these blood stages can kill people. In children, they tend to get stuck in the brain tissue and can cause cerebral malaria. But this is the stage of malaria that kills, the blood stages. While all that's going on, that clever parasite is anticipating that that person is going to be very sick and he's going to get cured or he's going to die. And so that parasite is already getting itself prepared to be transmitted to a mosquito. It hives off some of that normal life cycle to stages that can be picked up by the mosquito and grow and then return to be sporozoites. For our vaccine, we take those sporozoites and we blast them with irradiation, and that does a thing called attenuation. It stops the parasites from growing. So if we inject the parasites, they get to the liver and they stop. So you don't get any parasites in the blood, you don't get any disease, you don't get any transmission to the mosquito. And if we get the right immune response in the liver here, any infectious sporozoites that come in They're killed in the liver. That's it. That's what we're trying to do. So here's that life cycle, and what we've had to do is to try and make a malaria vaccine from sporozoites, because we know that they can protect. You've all had mosquitoes in your garden. You've seen them growing, the larvae growing in buckets of water and so on. They are filthy things, right? They're really dirty. You can't go putting something covered in bacteria, injecting that into people. So we had to come up with a way of growing those mosquitoes in what's called aseptic conditions, meaning that they have no bacteria, no fungi, all the way through their life cycle. And we succeeded in that engineering issue, in that problem. And we've basically taken that life cycle and made it linear. And we now grow the parasites in blood that we get from a blood bank. We feed them to mosquitoes that are aseptic. And we let those mosquitoes incubate the parasites until we get sporozoites. We blast them with a radiation. And then we dissect out the salivary glands which contain the sporozoites and purify those sporozoites, cryopreserve them, meaning that we freeze them at ultra low temperatures, but they're still alive. They still come out and they will infect people and so on, and then do all sorts of quality control on those. That makes it sound rather simple when you put it like this, but the research and development to get this far was about 5 years of nothing else. It was really a long, long slog, and you're never quite sure what is going to give you the issue. So at one point, we were trying to make aseptic mosquitoes, and they wouldn't turn into pupae, and then they would turn into pupae, and you wouldn't get adults, and then you get adults, and the adults wouldn't fly, and then the adults would fly, but they wouldn't feed, and we just had to keep playing around with these things until everything worked. Then we'd start feeding the mosquitoes on the parasites in blood, and the parasites weren't very effective, and blah, blah, blah. And we went through 6 months of that and found out that the incubator we grew the parasites in was slightly off, and all these things happen. It's a tremendous amount of things that you have to deal with. And it's the same thing. Things will go wrong, but you can fix them. So what we end up with at the end of all this, let me keep an eye on the time here. How am I doing? Have I got plenty of time? Yeah? So what we end up with at the end of all this research and development is what we have as platform technology. There's this wonderful mouthful here, which is aseptic, purified, vialed, cryopreserved, Plasmodium falciparum sporozoites that meet all regulatory standards. Hands up all those who understands what that means. I don't blame you. Right. Okay, so let's just go through one or two. Aseptic means they're without germs, right? There's no bacteria, no fungi, as much as we can tell. They're purified, because we want to get rid of all of that mosquito salivary gland tissue, because that has a lot of allergens in there. They're put into vials, and then they're cryopreserved, which I told you about. It's that species of malaria, Plasmodium falciparum. It's the sporozoites. And when we say that they meet all regulatory standards, essentially everything we do is reviewed by the FDA. They look at everything that we do, everything that goes into that vaccine, the fish food that's used to feed the mosquito larvae, where the blood comes from that we feed the mosquito adults. Are we using the right plastics for growing the parasites? Do they have leachables that could affect people? The FDA review absolutely everything, and we have to meet those standards. And then all these things then, all these different vaccines that we're working on are all grown in the same way. So this is a vaccine that's irradiated. We have sporozoites that we keep completely infectious so that we can infect people. And that's a whole other story. If people want to hear about it, I can tell you. But we have actually taken our infectious sporozoites back to Africa in several studies now and infected people to try and understand how malaria immunity works. If we give these sporozoites to the people at the same time as they're taking malaria drugs, we can get very, very strong protection. I'll show you a little bit of data on that. And then we're working right now, we start a project in about 3 weeks' time on parasites where we've knocked some of the genes out of the genome so that the parasite won't grow anywhere beyond the liver. So all of those are manufactured in the same way, but they have different outcomes. So what do you need to do to get a vaccine license? The first thing is always safety. There's no point having a vaccine if it kills somebody or if it makes them really, really sick. People won't use it, and people shouldn't use it if it's dangerous. Then it has to be very tolerable. You have to be able to say that people will come back for their second dose and their third dose. It has to not give lots of pain when you inject it. And then, of course, it seems like it's third on the list, but it has to be third on the list. It has to work. So what I'm going to try and show you very quickly is a little bit of information about all three of those things. And what we've taken, just with all our friends in the consortium up there, is to take a different strategy in the way we're developing this vaccine. Hands up all those who had the flu vaccine in the last couple of years. Yeah. Anybody also get flu? Yeah. It doesn't work very well, does it? Right? But it is safe and it is tolerable in most people. I know it's not tolerable in one person. But it is safe and it is tolerable. And it has an efficacy last year against the circulating strain of flu, had an efficacy of about 30%. But it's still recommended, you still get 30% coverage, so you may be slightly better off with it. But what we're trying to do is to go beyond that, not just push a vaccine onto the market because it's there, but is to get the maximum efficacy that we can before we get it onto the market. And that means that we are working with all these people up in the consortium there on different studies that will get us to a point of efficacy. What was one of my first take-home messages for today? Things will go wrong, right? Our very first vaccine trial we did just down the road here in Bethesda, and we injected our vaccine at increasing doses, and guess what? It didn't work. We knew this vaccine should work. All the history of it told us it should work, and it didn't. It didn't work, and we couldn't figure out why. And we were worried that we're over-processing these sporozoites, that they wouldn't work. In the end, as part of this study we actually injected some of our vaccine into monkeys. And without going into the detail, in our vaccine study, we injected our vaccine into the skin. If we took that same vaccine and injected it into monkeys, in the vein versus the skin, we saw completely different immune responses. And the results told us that we really needed to change the way that we inject our sporozoites. And so we did a second study in 2013, and there we got fantastic protection. We actually managed to have everybody in one group completely protected against malaria. And just as an aside, the way we do these studies is we vaccinate people, we let the immune response develop, and then we take some infected mosquitoes and we feed them on people. And the way that we got the protection in this case was to give the sporozoites intravenously. And that led us to develop a technique which we call direct venous inoculation. And really, it's just copying the mosquito. So here's a couple of movies here. We're both shots. Yeah. So this is the proboscis of a mosquito. You can see the mosquitoes feeling her way into a capillary. And this is us injecting our vaccine into a vein. We were a bit slow, but we were also very cautious because it's a new vaccine. We needed to copy the mosquito because she has evolved to do this over millions of years, and we're doing it from scratch. And the intravenous injections made a tremendous difference. But the problem with all of our work really was that all the immunity has taken place here in the liver. How do you measure what goes on in the liver? You can't. unless you're one of these guys, and you can cut somebody in half and have a quick look inside and figure out what's going on, you can't measure what goes on in the liver. And a lot of what you would normally do with a vaccine, like anybody get hepatitis A vaccinations? I do. I get them because I work on blood a lot. And how do they measure your hepatitis A reactivity? Do you know? They'll come back and they'll take a small blood sample, I think 6 weeks after the last dose, and measure your antibody titers. You can't do that with our vaccine because the antibodies aren't particularly important, and all these T cells that are important are taking place, doing their job in the liver. Very, very difficult to measure them. So instead what we've had to do is to come up with a hypothesis that said if we can get the dose right, if we can get the dose regimen right, meaning the way that you present those doses, we can go through a series of thresholds in protection. Because protection is the only thing we've got to go on. We've got nothing else, really. So we can vaccinate and we can get protection for a short period against the same strain of parasite that's in the vaccine, then for a short period against different strains, then for a longer period against the same strain, and for a longer period against different strains and wild populations of parasite. And that's the approach we've taken over the last few years. It's all based on this hypothesis here. And so we've gone to getting really good protection with last couple of years ago with intravenous injection. From there we've gone on to a set of papers that came out this year that were really high impact. We showed excellent protection against these biting mosquitoes all over again, and we showed with increasing the dose that we could get protection in people for 6 months against the same strain of parasite. So it's getting closer to where we wanted to be, but protection against a different strain of parasite was not that great. We've managed to show in another study that we can get protection right there, can't speak anymore, right through for a whole year, over a year, which puts us at the point where we're getting close to a vaccine that we could use for elimination in a focal area in Africa. If you can prevent people getting malaria for a whole year through a whole set of transmission seasons, you have a chance of doing something really important. And we also managed in Mali, in West Africa, to demonstrate that we could get significant protection. It's not at the level we want, but it is important. Now here's another example of not giving up when things go wrong, because before we knew the results of that study in terms of protection, we looked at antibody responses in our volunteers. And at the same doses, these in green are the ones people are protected, and red are the ones not. But we looked at all these antibody responses in Mali, and they're way down here, way down. And in our US volunteers, they could be really sky high. And we're thinking, oh, we're not going to see any protection here at all, because of these terrible looking results. And in the end, we got pretty good protection. Oh, let's go. We actually saw 50% protection over 6 months. So 50% of the volunteers in that group over 6 months were protected, and in the controls, 93% of the volunteers got malaria. Now, I'm not going to go into the subtleties of how you measure that protection. You could say there are different ways of doing that. But it's a really, really encouraging result. It gave us a lot of hope, and it's also opened up. I guess another thing that happens is when things go wrong, they sometimes end up going more right. So our first vaccine trial made us have a radical rethink about how we were doing things, and results like this, where the antibody responses are so low, has triggered a whole set of discussions of which Jongo is a central part, about what the hell's going on? What are the immune responses here? What are the people in Africa doing who've had malaria many times? How are they suppressing their normal immune responses and still able to get protection with our vaccine? It doesn't make intuitive sense. And it's triggered a whole lot of really interesting and stimulating discussions in our consortium and further afield too. We are, of course, paying a lot of attention to different strains of malaria. Can we protect against different strains? And we've shown that we can get good efficacy for up to 8 months, but it's still not quite enough yet. And at Jongo's institution in Tanzania, it's a place called Bagamoyo. It's on the coast, just north of Dar es Salaam. A lovely place. It really is very nice. We did our first vaccine trial on children, the first in Africa, first anywhere on children. And you can see that this mode of injection into the vein is still possible on very, very young children. When everybody said there's no way you could ever do that. And your nurses are finding it rather easy now, right? They're really brilliant, they really are. So Tanzania led the way on that study, and now we're in the middle of a big study in Kenya, where the malaria rates are probably some of the highest in the world, with 330 infants getting either the vaccine or the placebo. And almost any day now, or we just started actually with children in Equatorial Guinea on the other side of the continent too. So we're really working hard to get our vaccine into the people who need it, which are the children. They're the ones who really suffer the most from malaria. Here's our volunteers with their mums. You're not allowed to show the faces of volunteers, so that's why they've all got your back to you. You're not on this picture, are you? No. Sorry, I'll find a good one of you. So without going into data, this is how we summarize the trials, but you can see that we're getting high levels of protection here in the US, both short-term and long-term, and 50% protection at this stage of the game, where we're still in what's called phase 2 clinical trials, is actually pretty good. We're happy with it. We've got a long way to go, but it's really pretty amazing that we've managed to get so far in just 8 to 10 years. Now, in some of those trials, we have what's called a double-blind placebo-controlled trial. And essentially what that means is that the person injecting the vaccine, which is usually Jongo and his team or equivalents elsewhere, don't know what's in the needle. And they will inject either the vaccine or saline, just the same saline as you would get in a drip at the hospital here for rehydration. It looks identical, you can't tell it apart, and it's only after 6 months, 9 months, a year, maybe longer, when you break the code of those volunteers, do you find out who had what. All the time that you're going through this trial, you're collecting data on those volunteers. Were they ill? Did they have any adverse events? Did they feel sick? Were they sick after getting vaccination? Were they having more headaches than normal? And then at the end of the trial, you unblind all those data and you look to see if there's any difference between the people having the vaccine and the people having the placebo. And in every trial we've done to date, you can't tell the difference. It's a remarkably well-tolerated vaccine. And we think that the reason for this is partly because there's no adjuvant in there. An adjuvant is something that is used to stimulate the immune system, and by very nature it's irritating because you're trying to irritate the immune system. Another reason is that we're injecting something that has evolved over millions of years with people and with other animals, and so it's developed its own way of being non-stimulatory. So it doesn't aggravate the immune system. It wants to get into that liver, it wants to chill out in the liver for 5 days and then grow. Then it gets nasty. But it doesn't want the immune system to be aggravated for those first 5 days. And so it's a really well-evolved system. So that we're more than happy with that. It's incredibly well tolerated. And then when we inject it into the vein, the volunteers don't actually record any pain. In Ifakara, in Bagamoyo, the thing that was really irritating the children the most was not the injection usually, but the tourniquet being put on. And it was pinching the skin and so on. And one of Jongo's colleagues came up with this wonderful idea, which worked beautifully. 10, 15 minutes before the kids came in the room, he would go outside and play with the tourniquet. And the kids just put it on, take it off, play with it. They could throw it around a bit. When they came in, all that stress had gone and the kids were much better. And we actually had one baby that was feeding on the breast of his mother and didn't even wake up when we put the needle in. These needles are so small and sharp that it's hard to tell. So this is what happens in a typical field situation. This is our injection team in Mali. I'm on the other side of that wall somewhere, and we pass the vaccine through. And this is a young physician in Mali who, I apologize, I can't remember his name. You know, one of the volunteers gets sat down. You clean them up with some antiseptic of some sort. You can use alcohol. And during this time, or just before this, he's checked to make sure that this person has got a good vein available. Clean it all up. And meanwhile, the vaccine is sitting there in this needle and syringe in the tray over here. The tourniquet goes on. And this is the bit the babies really didn't like. They didn't like it very much at all. You can see he's telling her to make a fist. Finds the vein. Bang, in it goes. See the blood flash. That shows, being able to suck that back says that he's in the vein already. And in it goes. Really painless and very, very easy and very quick. And so many people have told us that we couldn't be able to do this. And we've done it now in the, I can't think how many thousand volunteers. So we, as a response to that negativity that we get, we've asked people when they've been injected, did you get any pain? And most people said no. 97% of the people said, they had, or 97% of the events, they had no pain at all in 2 or 3 different studies. The other reason why it's pain-free is partly because the needle is small. There's no adjuvant. You're not putting it into a muscle and you're leaving that bolus of fluid in the muscle, and it's painful. And so it's just going straight into the vein and gets dissipated. Now all of what I've talked about fits into a grand plan, believe it or not. It sounds like we're just chugging away quickly and getting on with stuff. But we're trying to go through a set of phases of study, and we're in the midst of this stage 2 at the moment. And our focus right now is on optimizing the vaccine, getting the right dose, getting the right combination of times and numbers of sporozoites in the dose and so on, so that we get the best protection possible. And sadly, because all this stuff with not being able to sample the liver and so on, the only way we can do this is with efficacy studies to test whether it's actually worked or not. And at the same time, we have to think of making this stuff. So we have a clinical manufacturing facility, which is just 5 minutes walk from here, where we make the vaccine. And as you go through these stages, the FDA requests better and better control of the way that you manufacture your materials. So we're going through stages at the moment where we're upgrading our facility. And we have to then do very big trials in Africa and the US and so on. I think we'll skip through that. Now, one of the good things that happens here in the US compared to almost anywhere else that I know is that the FDA can be very actively involved with you. A lot of people say the FDA are a bit like the police and so on, and they're in the way. But actually, if you work closely with the FDA, they can be really, really helpful. And we talk to the FDA all the time, and they have a wonderful turnaround process. You know, this is probably the biggest regulatory agency in the world. They still guarantee you a response in 30 days. Nimri? He's laughing. So the equivalent in Tanzania, it goes into a black hole sometimes, doesn't it? And they say 2 months, and then it's a bit longer and a bit longer. And so it's a very flagship organization, I feel. But the other thing that the FDA have done is to stimulate research into neglected diseases. They'll give, when a research and development gets to a certain point, they'll give all that research and development fast-track status, which means that instead of taking a month or 2 months, they'll pretty much halve the time. Every time you ask them a question or need a response or documents reviewed. And that was given to us last September. It means that we'd reached a certain point with our research that the FDA thought it was worthwhile prioritizing what we do. So that was very helpful and more than satisfying. Now remember at the very early on I told you about having infectious parasites being given at the same time as malaria drugs. That's what we call CVAC, right? It's chemoprophylaxis vaccine. And I lied to you at the start. There is another technical slide here I'd forgotten about. This slide here represents the malaria life cycle in the liver. So the sporozoites come in, they chill out, then they find a liver cell and they'll invade. And if it's one of our attenuated parasites, it stops right here, and the immune system attacks it, and that's our vaccine. With the infectious sporozoites, they go in, they grow, they multiply 40,000 times, come out in the bloodstream and do their stuff and kill people or whatever. If you've got chloroquine, a malaria drug in the system at the same time, those blood stages form, you get one set of parasites going into the blood and then they're killed by the drug. And for some reason, that induces an immune response that is really, really good, really protective. And we are able to induce an immune response with about 10 to 100-fold less sporozoites, fewer sporozoites, than we would with the vaccine. Because probably, not only do you have the parasites around for a longer time, you've got more of them because they're multiplying, and as they're multiplying, they're changing their developmental path, stage rather. And so you've got a bigger diversity of antigens for the immune system. So again, we managed to show that we can get really high immunity and really good protection with this approach. And we're about to start more studies on that in Ghana and Equatorial Guinea. The interesting thing about this approach, which we're a long way off this, but we, I guess if nothing else, we're pretty aspirational at Snanaria. We like to think ahead, is that you can think about giving these infectious sporozoites at the same time as you would give a drug for mass drug administration to cure malaria in a whole area. So you'd come into a village and give everyone the drug, and at the same time, if you could give them the sporozoites, you'd cure them of malaria that they have at the time, but you'd also give them highly protective immunity from getting malaria again. That's what we're thinking towards. It's a long way off yet. I think I'm going to stop there. I think, or will I? I want to come back to people again. So I was talking about being aspirational, and I talked about Marcel Tanner very early on. Marcel is a very practical guy, and he doesn't like to see research just keep going and keep going and keep going. He'll look you in the eye and say, when are you going to do something with that research? When are you going to turn it into a vaccine? When are you going to actually use it? And he's been, the last week or so, while he was in, unfortunately I was in the UK when he was here, but he's bugging Steve Hoffman saying, let's get a program together where we test this vaccine properly. And he's very much into testing effectiveness and efficacy. And on this island called Bioko, which is part of Equatorial Guinea, has a population of about 250,000. we're going to be testing whether we can eradicate malaria with a vaccine. And again, just to come back in a big loop to people, this gentleman here is the late D.A. Henderson, who is like a giant in the vaccine field. He led the charge to eliminate smallpox. He was the guy who was out there rolling his sleeves up in India, finding out where the last bastions of smallpox was hanging about. He helped to develop this method of immunization, which is the scraping on the arm. I think the few older ones of us in the room may still have scars from that. And he came to Snanaria, he met with our kids, and he's really inspirational. He says, you've got to try this. You can't just make a vaccine and hope that it's going to get out there all by itself and someone else will do it. It's your vaccine, you push it. And he, until he passed away last year, he was on Snanaria's advisory board and gave us some very sage advice about how to get things done. And the way we're going to get things done is to demonstrate it in a place that's quite restricted so that we have control over it all. And that will be hopefully on this island of Equatorial Guinea in a few years' time. That said, we keep trying to innovate. Now, remember I showed you right at the start just how complicated our vaccine development process is and making things in mosquitoes and so on and so forth. Let's just go through. Instead of making things in mosquitoes, what we're working on very heavily is to get rid of the mosquitoes altogether and grow the parasites in a test tube, basically. And we're having some great successes with that. What you're seeing here are sporozoites that have been put onto liver cells in vitro in a test tube and shown to grow whether they come from the mosquito or from cultures. It means that we are getting close to being able to grow our vaccine in huge quantities using robots. The price goes down, it's just amazing if we can get that to go. It's just a game changer, and it'll bring the price of the vaccine plummeting down. There's things that we have to do in the field. For example, we have to demonstrate that our vaccine is safe in people who are more vulnerable. One of those groups are people who are HIV positive. And Jongo is going to take the lead on a study in there very soon in Tanzania where we'll test it. And we will also have to test this in pregnant women at some point, and we're working on plans for that too. And then we need to be able to say, all right, all of everything I've told you about is all about one species of malaria. And there's actually 5 species that infect people. And Plasmodium vivax actually infects more people than falciparum malaria, but it's not so dangerous. So it doesn't get quite the same attention, but we have to develop ways to challenge people with vivax malaria. So we've got lots of things going on now. We're going to have to make a multi-strain vaccine, and that has all sorts of nightmares of its own. We're just about to start that actually next week, I think. We've got to improve our protection, and then we've got to do all sorts of other studies to get to where we want to be, which is to be able to give this vaccine to people and reliably get protection against malaria. And finally, it is all about people. And these are all the people at Snanaria that are doing all the work. I get to come out and cheer for them now and again like this, and to excite people, hopefully. But every one of these people just is just ridiculously committed. I've never worked in a place like it. So with that, I will take any questions that you would like to throw at me. Thank you.
How do you get people to volunteer when you're at the beginning of your trials?
it's actually not that hard. It's harder. So there are all sorts of groups of people that volunteer. One of them is medical students. They tend to volunteer quite often for things. In places like Baltimore, they actually have people who volunteer for everything, and that's the way they make some money. The way that you get people is through advertising. If you look at the back of the Metro magazine on the train, you'll see adverts in all sorts of clinical trials in NIH. You'll see posters. You'll sometimes hear it on the radio, people advertising for these campaigns. And then the people that lead the studies for us at the clinics, such as at the NIH or in Ithaca or anywhere, they very often have a network of people that they've pre-screened who have already put their hand up to take on a trial, and when ours comes along, they'll review it. What we're not allowed to do is pay people any significant amount of money. You're allowed to give money that covers their time for lost doing something else. So it's you don't get a great recompense for it. Or if you're a young student, you might think you're getting a great recompense. If you're a working adult, you think, maybe not, I'm not interested in that. But so far, with very, very few exceptions, it's not been difficult to get people to volunteer. In Equatorial Guinea, our issue was not getting the volunteers, but was getting healthy volunteers. So they were coming in and they were having to be excluded because they had diseases that could interfere with the way that we interpret the results. So they'd have diabetes or kidney malfunction or serious worm burdens and so on. So there in Equatorial Guinea, to get particularly the older age groups, it took us a long time to get enough volunteers. Again, not because they wouldn't come through the door, it's because they weren't healthy enough. Maybe in Africa they have a slightly different system where there's a very strong community engagement up front. Dongo, tell me if I get this wrong, but generally there's a priming of the community that this vaccine trial is going to happen, and giving information out about it for a long time beforehand. And then when the vaccine trial begins, then there's a more serious recruitment goes on, and then people come into the center to get recruited and enrolled in the study.
Do you encounter any ethical roadblocks in doing this? I'm wondering, especially with having people infected with malaria?
Yeah, so the infection of people with malaria by mosquito bite has actually been going on for about 40 years, maybe longer even, for all sorts of reasons. And way back, it was actually used, malaria was used as a treatment for syphilis. Before you had antibiotics, people would get malaria, and the immune responses against the malaria were so profound that they would partially or completely cure the syphilis. And then there are a lot of studies way back on irradiated parasites, where instead of giving it through a needle and syringe, they would give maybe 1,000 mosquito bites to people. So all of the ethical issues surrounding that, fortunately, were dealt with before Snanaria was founded. And then as we moved into Africa, Jongo's boss in particular is one of the people that says this is an African disease and we want to be taking the lead in working on this disease. And he says that because there's been this reluctance for Western vaccine developers to work with Africans as a front line, right? Is that a fair judgment? Because they felt that we were taking advantage of undereducated, resourced populations, and I think that's going away. We feel that we give a really good instruction to people, and it's frontline. So there were ethical issues around that, and then we were told there would be ethical issues about injecting directly into the vein, but so far, no one who's involved with it has looked at this and said there's a problem. And so that's also gone away. So these things do arise periodically, and in every vaccine trial we do, we have an independent ethical review committee who, depending on the trial, they'll be more or less involved. So, for example, excuse me, in the first trials we did with children, we went very, very conservatively from adults to older children, teenagers, then to middle, and then down in age until we got to where we wanted to go. And at each step, the ethical review committee would review all the data, all the safety data, and say it's okay to proceed. And they're an independent body that we work with. And so that sort of oversight from very wise people, very experienced in the field, is really very useful to make sure that you address the ethical issues before they actually arise. It's really helpful to have that.
So have there been any issues with vaccine of those strains?
Just cost and hard work. We work with a strain called NF54, which is the equivalent of a weed. It grows in the lab really nicely. You can infect mosquitoes really easily with it. You get reproducible results time after time after time. It's the only strain that does that. All the other strains of malaria that you can grow in the lab often don't infect mosquitoes at all. And then if they do, it's unreliable and it's low levels and so on and so forth. And until we started working with one other strain, which comes from Brazil, we even now we don't feel that our results with it are that consistent, but we've got them better and better over the years. So it's really just the hard work of it. It'd be much nicer if it's a single strain. It ain't going to happen, I think. Any other questions? Okay, in that case, I guess it's pizza time for you guys, right? Caleb, any other things that you need to?
Sure, yes. What would you say is the number one limitation to not just your job, but just field of research in general? So if someone is torn between becoming a healthcare professional or going into graduate, there's obviously lots of benefits, downsides on either side. What would you say are the main benefits and also the main limitations, downsides for postgraduate?
So I'm going to be very biased in some of these answers, but for me, the big benefit about going into research is the excitement of it. And if you get the chance to do what I did, you can call the shots. So all of that work that I described when I was an academic through London and Scotland and Switzerland, I wrote the grants for those, and I defined what I wanted to work on, and as long as I got the money to do it, I could do it. I could spend my time in the field in Tanzania, and I could work with the deer in Scotland and so on because I got the grants to do it. So that's the exciting side of it. The flip side is you've got to get the grants. And they can be very, very competitive and very, very difficult to get. So if you don't just get to do research, it costs money. And to get that money, you have to justify what you want to do to an agency, such as the NIH, or in the UK they have a thing called the NERC, which is Natural and Environmental Research Council. You have the agricultural equivalent of it. There's so many different bodies that you can go to get money. But if you're not good at writing those grants, and if you can't justify what you want to do, you can have a very, very barren research project, right? You'll just get the minimum money, and you'll be basically paddling through treacle, through molasses. It's awful if you go through a phase where there's no money, so that's a downside. If you're moving into the clinical side of things, God, yeah, you get paid better, right? If you're going to become a physician or something, you can do the same job as me and probably earned about $60,000 a year more per year. Oh, hello. Hello. So, and then as a physician, I think you have more avenues open to you. So it's much easier to be, let's say, a physician and be interested in entomology, which I've met several people like that. So they keep their physician role and then they can be working on mosquitoes or bugs or whatever. But you can't do it the other way around. I, as an entomologist, I can't go and stick needles into people and diagnose them with malaria. That's against all the rules, right? So they're probably, if you go into the medical side and work as a physician, there's certainly more doors open to you. But it is a longer and harder road to tread as well. Much more competitive to get there. But once you're there, I think there are more doors open to you.
Were you ever torn between, professionally or?
I never wanted to be a doctor until it was too late. So I think the only time I really thought about being a doctor was while I was at Imperial College, and I flirted with that for about 10 minutes. And I thought, if I do this now, I stop this beautiful research program, and I have to go to university for 5 years, and I have a child, and I can't afford it. I can't do it. I've left it too late. I probably hadn't left it too late. If I really wanted to do it, we would have found a way of making it happen. But I never really wanted to go that direction, to be honest. I love biology. It's much more fun than medicine. Right, Jonga? No answer from the front. Any other questions? Okay. Another one?
I need to ask all the questions, but so you did your studies internationally, not in the U.S. Would you have anything to say about the benefits of pursuing an education in an international setting?
Yeah. Yeah. So some real pros and cons. I guess I've been involved with academic institutions in 4 or 5 or 6 countries in one way or another. The UK system, meaning England and Wales, you get locked into a degree pretty much from day one. And it's a 3-year degree, so you have 1 year less of fees and you'll reach your bachelor's degree quicker, but you've got almost no flexibility in the system. It's very hard to change. If you don't like what you're doing for some reason, it's difficult to shift over. In Scotland, it's a little bit more like the US, where you have a bit of flexibility, particularly in the first year. The US I like much more in many ways because you do have that ability to continue your broad education for another year or two. But I personally don't feel that the end product from an American degree is as in-depth as from a UK degree, but the breadth is way better, way better. And you can get the depth later on. So I guess I'm saying I sort of prefer the US system slightly. For the PhD, again, the UK system is very contracted. It's 3 or 4 years, whereas in the US, it tends to be at least 4 years and typically more because you have coursework. And in the UK, you tend not to have coursework. It's 3 years, welcome, introduction, bit of orientation, get on with your research project. See you in 3 years' time, right? Very, very different. Very much get on your feet and get on with it from day one. There's not a lot of hand-holding and hugging goes on. It's very straight at it. And part of the reason for that is tradition, and part of it is that we have in the UK 3 years of money. If you get a research grant for a PhD, it's 3 years, done. No extensions, nothing. So you're expected to get it done. If you've got field work in there, if you're going off and doing an ecological project in a place where there's seasons, you're lucky if you get 2 full seasons in, right? And that's, data gathering wise, that's not brilliant either. So again, in the US, I think the relaxed nature of the PhD, or I doubt there's a PhD student here who call it relaxed, but it's a more relaxed program and timetable, is better in some ways. And but it means the difference on the flip side again is that you come out here older and in a lot more debt by the time you finished a PhD in the US compared to the UK. I'd say you probably get about 4 times more debt here than you have as a European student.
Are the UK programs competitive?
Meaning to get into?
Well, exactly.
Oh God, yeah. I mean, I used to be in charge of the graduate school at Aberdeen University for the life sciences, and we would have approximately 10 to 15 applications per PhD.
So if you were to give some advice to some middle school or high school students or even undergraduate students about what kind of an earlier stage in their educational career, what advice might you
Oh
give?
God. So, I mean, you've always been told to find things that make you stand out and look a little bit different. And I'm personally a big fan of, partly because of my PhD, of people who've got some outside interests. I'm a sporty person, so I tend to like people who are sporty, but that's really not the big deal. I think you really have to find a way of communicating your passion for biology or whatever it is you're going into. You have to find some way of communicating that either in an interview or on paper. And the best advice there is don't just throw your application into the ring. Get people you trust to be really critical of you to read your application. That goes for anything in life, actually. If you're writing your CV and you're going to trying to get a job at a local store, I would still get your mum and dad to read that CV, and I'd still get your teacher or somebody else that you trust, or one of your parents' friends who's really high powered, and say, look, here's my CV, is it okay? Here's my resume, what do you think? And get used to having that interaction with people that you know can give you feedback and give you honest critique. So they don't just say, oh, that's really nice, Johnny, that's a lovely little resume there, and they're thinking, God, that's awful, right? So I'll give you an example. My eldest son, when he was trying to get into medical school, he wrote his essay that goes with the application. And it's a 1-page essay, and it's, why do you want to be at medical school? And I was traveling, I said, why don't you send it to my sister-in-law, right? Because she was the dean of nursing at Sheffield University at the time. And she sent it back to me, not to him, and she said, I don't know Michael well enough to tell him that this is awful. And I said, tell him, it'll be okay, I'll be there when you tell him. And she was on the phone for him for just 10 or 15 minutes and said, Mike, you've got to do a lot better than this if you want to go into medical school. And it was brilliant for him. She didn't hold back. She's a tough woman, right? She's really knows her stuff. And she said, if you want to be competitive with all the other people, do this, do this, do this, do this. So I think more advice you can get from people who know what they're talking about, the better. And we all do it all the time. I still do it now. I still send things to people to read. Good? He's falling asleep here. This poor guy is tired. Well, thank you everyone for hanging in there. It's been really great. Thank you very much.